Hi, I'm Christian Stevenson with the Mississippi State University Extension Service here in Hancock County. I'm going to be joining you for the next a little while talking about plant propagation. And this is a very broad topic, a lot of sort of sub areas that we can discuss. I'm going to try to just give really an introduction to the various kinds of plant propagation. There's a lot more detail that we can go into, hopefully, in future presentations. And I'm going to start off talking about propagation by seed. Sometimes you'll hear this being called sexual propagation to contrast it with other forms of propagation. And we're fortunate that plants tend to produce a large number of seed. The advantage of using those seed as a form of propagation, firstly, is that it tends to be relatively inexpensive if we're purchasing seed, or we tend to be able to gather a fairly large number of seed from plants, so we can propagate that large number of plants fairly quickly. Sort of the other side of that is because we're getting that the combination of those gametes, the male and female, we get a genetically distinct individual, and that allows us to maintain the genetic diversity that is there in the plant population. The new plant is going to have some combination of the characteristics that it received from its parent plant, and having that diversity in a population can maintain what we refer to as the fitness of that population. And where we use that term, what we mean is sort of the adaptability or the adaptation of that plant to the ecosystem that it lives in. Oftentimes, having that genetic diversity there can be a way that the plant species can cope with a disease. Some individuals may be more or less resistant than others. Just to briefly hit on another very broad topic, having that genetic diversity allows us also to select for qualities in a plant that we particularly enjoy. And the example that I like to use from that is Brassica oleracea, wild mustard. And you can see there that by selection for different qualities in the offspring, we can over time select for fairly expansively different qualities in those offspring. So everything from cabbage to broccoli to kohlrabi all arises from that genetic diversity. Now, when we collect seeds, we do want to keep in mind that we need to make sure that we are collecting mature seeds. This is, of course, most often an issue when we're talking about vegetable plants, because often we will harvest some vegetables prior to when they're actually fully mature. We also want to make sure that we're collecting seeds from healthy plants. Either we have an instance where some types of plant diseases can be transmitted from the parent plant to its offspring. That's relatively rare in a lot of cases, but another issue is that those parent plants may just be particularly susceptible to that plant disease. And the other sort of part of that is that we want that plant that we're collecting seeds from to be healthy, to be able to put a lot of energy into those seed and produce really good seed. I've already mentioned genetic diversity, so we do want to make sure that we aren't collecting all of our seed from one individual plant in the population. It's a good idea to collect seeds from several different plants just to maintain that diversity of the plants that we have in our landscape or in our gardens. Now, when we're talking about collecting seeds from fleshy fruit, and this again would be things like tomatoes or cucumber, a lot of our garden plants, removing those seeds Out of that fleshy fruit, oftentimes they're going to be in a matrix and in some of that vegetable matter. And we want to even keep that vegetable matter with it. Let that sit in a container for several days. Oftentimes, after it sits, it may form a little bit of mold on the surface of that, and that's perfectly okay. We can go ahead and take that mold off, add a little bit of water, just to kind of help us separate the seed from some of that plant matter. And we want to pay attention, generally speaking, with these seeds. If they float onto the surface of the water, that means those seeds aren't going to be viable. They aren't going to germinate and produce a new plant for us. So the seeds that we want to collect are going to be the ones that sink down to the bottom. So we want to pour off that water and collect those seeds. Now, those seeds are going to have a lot of water on them, and we don't want to store them immediately. We want to put those out on a plate or a piece of cloth or a piece of newspaper, out of the direct sun and away from any heat source, and just let them slowly dry. Make sure we stir them every now and again, just to make sure that they dry evenly. The reason we want to keep them away from direct sunlight and away from those heat sources is we want to make sure that they dry slowly, so that when they dry, they don't crack. As if they dry too quickly, that can actually damage the seeds. It's a little bit more straightforward collecting dry seeds, and this would be things like beans, primarily for our gardens, things that are going to produce their seed in pods, and we can allow those seeds to just dry on the plant. One thing we want to watch out for is oftentimes as those pods mature, they're going to crack open for those seeds to fall out. So it's a good idea to place a bag over the seed head or over the pod as they mature. And that way, if they do crack open and those seeds fall out, they're not lost, they're just collected there in the bag. A lot of times when we are collecting those seeds, they'll have a little bit of other dry plant matter with them. And you can use screens in order to sift that out. What I will normally do, because I'm working with a very small amount of seed, generally speaking, is sort of hand pick them out of that material. But if you have a large number of seed that you're trying to collect, just having a couple of screens will allow you to do that much more quickly. So when we're collecting seeds, we want to be mindful that the plant that we are going to produce from that seed is going to be what we're looking for, and how that plant is going to be pollinated is going to have a significant impact on whether it's going to be pollinated by a plant of the same variety. In the case of airborne plants, and a good example of that would be corn. Corn pollen is airborne, and it can blow for a significant distance. And so it can be difficult to ensure that we're not getting pollen from other plants. So if you have another variety of corn or another variety of an air pollinated plant, within a mile, it is possible that you'll get some air pollination because of that. In the case of insect-borne plants or insect-borne pollen, which is going to be a very large number of the plants that we work with. You can see on the chart there the fairly broad diversity of vegetable plants that have insect pollination. Those insects can move a pretty good distance between plants when they're out pollinating. And so we may get crossing with different varieties that are as much as a quarter of a mile apart. In the case of self-pollinated plants, and the classic example there is the tomato, you will generally get pollination by that same plant, so it's a little bit easier to manage whether you're going to get the same variety giving you that pollination. Though it's important to note that even though we say these plants are self-pollinated, Oftentimes, of course, insects are visiting these plants. They absolutely will spread pollen from one plant to another, and so you can get crossing even on these self-pollinated plants. So in order to take care of this, we need to make sure we take some steps. If we really want to save these seeds and make sure that we're maintaining that variety, we need to take a step to prevent that cross-pollination. Particularly for insect pollinated plants, what we want to do is we want to bag those flowers to prevent access by the insects to those flowers. And there's several materials that can be used in order to do this. Just needs to allow for light and for air movement. If you have something like a plastic bag, that's going to create a greenhouse effect in there, and oftentimes that is going to kill the flower. So materials like mucilin, or there is a geotextile called Rime that is used for this, can be made into little bags and then placed over the flower in order to prevent the access by those insects to the flower. When we're doing this, we do want to make sure that we, of course, you're going to tie the bottom, or you can use a twist tie or a little piece of cloth to tie that bag closed to ensure the insects can't get into them. We want to be really careful to make sure that we avoid damage to the stem. And we need to make sure, of course, if we're talking about a self-pollinated plant where the male and the female parts of the flower are there, um, then it can still pollinate itself within that bag. But if we think about imperfect flowers where it may have separate male and female flowers, we do want to make sure that we are pollinating that plant prior to when we are putting that bag over it. And there's several, of course, different methods that you can use to do that. The sort of basic way of doing it is to take a cotton ball or Q-tip, moving it between the male and the female flower in order to transfer that pollen. Some people will simply take off the male flower and sort of touch the two flowers together in order to deliver that pollen to the female flower. The bag needs to be kept on the plant until we start to see the formation of the fruit. Once the fruit is formed, we can take it off. Though it is a good idea, we're very rarely going to be bagging all of the flowers that are on the plant. So it's a good idea to keep a tie or something around that stem or around the base of where that fruit is going to be, just as a reminder to us that that is the flower, or that is the fruit where we're going to be collecting seeds, because we know that's one that we took care of. Now that we have the seeds, we do want to make sure that we're storing them correctly. We do want to make sure that we're keeping them under cool and dry conditions. That is going to be how they're going to last the longest for us. We want to make sure, if we've had seeds that have been kept in really widely fluctuating temperatures or with the humidity going up and down, very often those seeds are not going to be viable any longer. So it's probably best to discard them, or at the very least understand that our germination rate on those seed may well be poor. And the longer we're storing those seeds, the longer we're keeping them, the more important it is to control those moisture and temperature conditions. Some ways that we can take care of our seed in terms of managing moisture. One thing that is very useful is we can store our seeds with but not touching a product like calcium chloride or the dried silica gel packets that you see there in the upper picture. And those absorb moisture and will keep that away from the seed. Store them in an airtight container where they're going to really be in a low humidity condition. And just to be sure, there are some seeds like beans and okra where they can be over dried, and I wouldn't recommend using these silica gel packets or other materials with them, because that can ultimately reduce our germination for those particular things. Um, We can also store our seeds at lower temperatures. You can store them very easily at the sort of the temperatures that we often keep our refrigerators down around 40 degrees Fahrenheit. I use a cooler that's a little warmer than that, but still within that 40 to 50-degree Fahrenheit range. And seeds will maintain very easily in those conditions. If we're going to store them really long term, if it's going to be several months or even several years, we can store seeds in the freezer down below 32 Fahrenheit. And as long as we dry them properly and make sure we're not letting any moisture in, they can be maintained there for a very long time. The picture down there at the bottom is of the Svalbard seed vault, and that is what they do. They store seeds at a very low temperature with the idea of preserving those against some tragedy where we would need to restore the seed stocks that we have. It is important if we have seeds that are stored at very cold temperatures that prior to handling them, we allow them to come up to room temperature. That will just allow them to just to kind of more gently come up to that temperature and make it less likely that we're going to do some damage to the seeds as we're handling. And again, this is for vegetables, but you can find similar resources for flower seeds and other plants that you might have around the landscape. And this just shows roughly how long we can store seeds under those proper cool, dry conditions. And you can see that does vary a little bit by the type of plant we're dealing with. So plants like corn, we can store those seeds for about 2 years. Okra, in sort of the same range. Peas for about 3 years, about the same for beans. But tomato seeds, as long as 5 years, and still very effective, good germination for those plants. While this is a good rule of thumb, it is certainly possible to maintain seeds for significantly longer than that. It's just important that we maintain them under good dry conditions. I have managed to plant tomato seeds that are as much as 8 or 9 years long, or 8 or 9 years old, and gotten very good germination out of them. So as we're talking about germination, we do want to make sure we're using a good media in order to do that. Ideally, we're going to use a soilless mix for that. So a material like peat or coir, or even perlite and vermiculite will be very good for seed germination. It's got a lot of water holding capacity. It really allows air movement down into that material and provides a really good medium for that seed to grow. In most cases, light isn't really a concern for germination in most plants. There are some small seeded plants that do require light in order to germinate. They use the light as a signal that it's time to start growing. And there are a very few plants, Vinca minor comes to mind, where those plants will only germinate if they are maintained in complete darkness. And one thing that's really important that I mention here is while those conditions, you may not need light for the process of germination of the seed, but immediately after germination, you are going to make sure or need to make sure that you have good light for those plants because you don't want them to get leggy as they're starting to reach out for the sun. Moisture, of course, when we're starting new plants, we do want to make sure that we maintain really good soil moisture. Plants are going to be really susceptible to drying out when they're very small. Temperature, generally speaking, a temperature range of about 75 to 85 degrees Fahrenheit is where we want the temperature to be in order for good seed germination. You can see that there's a pretty wide range of temperatures that will be acceptable, but what we want to look for is right in that optimum range. If you kind of scroll through all of those different entries, you'll see that just about everything is going to perform well right in that 75 to 85-degree range, where you're going to get really good seed germination. Another thing that we do want to note is that some seeds can have an issue we refer to as seed dormancy, and they need to be or stratified, or they need to have that dormancy broken in order for them uh, to germinate. In some cases, that is exposure to cold temperature. The higher temperatures doesn't allow oxygen to pass through the seed coat, so they need a period of time when they're under those cold temperatures in order to start to germinate. In some other cases, particularly where seeds would naturally go through an animal's digestive system or something like that, they do need to be exposed to a weak acid or sandpaper or some other material to sort of scuff up the outside of that seed coat and break that thick seed coat to allow them to germinate. One of my experiences dealing with this was working, trying to germinate kudzu of all things. And we would have expected that kudzu would germinate very easily given how widely it spread. But when we tried to do it, we just couldn't manage to get any of them to germinate. Did some research and came up with a method that we would have to use in order to germinate them, which involved soaking them in hydrochloric acid in order to break up that seed coat. All right, so that's what I wanted to say about seed propagation or sexual propagation. And now I'm going to move on and talk about vegetative or asexual propagation in plants. In order to do that, I'm going to talk a little bit about different kinds, including division and layering, as well as after that, talking about cuttings and grafting. What's really important when we talk about vegetative propagation is that we are using a part of that parent plant to produce a genetically identical offspring. So unlike where we have sexual propagation or seed propagation, where you have a completely new, genetically new individual, in the case of vegetative propagation, what we're essentially doing is producing a clone of that original plant. And that has some advantages and some disadvantages. We can very easily reproduce a plant where we really like its qualities and be sure that we're going to get more plants that have that quality. But at the same time, if we do have susceptibility to some problems, then we're going to see that same susceptibility in all of the plants that we produce this way. So the first type of vegetative propagation that I'm going to talk about is division. And this is really just separating off plant parts in order to produce new plants. And we do this with bulbs and rhizomes and clumps and suckers. And generally speaking, we are going to do this during the dormancy period of that plant. So starting off talking about bulbs, and that includes plants like hyacinth and lily, narcissus and tulips, as well as onions. Of course, naturally, bulbs will produce offsets next to the parent bulb. And as we are lifting up those bulbs for storage at the end of the year, we can remove those offsets and maintain them. If we have really large hardy offsets, we can just replant those into the ground immediately and let them be maintained. Sometimes with smaller offsets, it is better to maintain them in pots or something like that for a short period of time, just in order to allow them to build up a little bit more strength before we move them back out into the native soil. It's also important to keep in mind that when we're separating those offsets, oftentimes they aren't going to flower in that first year. They will put up leaves or vegetative growth, but oftentimes we won't see flower formation just because the bulb really needs to take in a lot more energy in order to be ready to produce those flowers. Again, the large hardy offsets can be planted right in the ground immediately. Small offsets, we're going to want to put those in pots until they've reached a bit larger of a size. One thing we can do if we're particularly, wanting to produce more bulbs, bulbs can be relatively expensive. So this is a good way in order to just build up the number of them that you have. You can plant your bulbs shallowly, and that will promote them producing those offset bulbs. Another thing we can do is notching the basal plate of the bulb, and you can see a sort of diagram of a bulb over there on the right side of the screen, where you see the scales of the bulb, and the basal plate is right down there at the bottom, where all the scales are coming out of and where the roots would be. You can just cut out a couple of small sections of that with a really sharp, really clean knife. And that will, again, promote the formation of those offsets so you can get more bulbs. Another action that you can take in order to produce more bulbs is scaling. And this is going to be for true bulbs that are formed out of multiple scales, things like lilies or fritillaria. Makes a very effective way to propagate them. In order to do this, you just snap off 4 to 8 scales from as close to the base as you can possibly get. You ideally want to get a little of that basal tissue with the scale that you remove, and then press that base end firmly down into a tray of sand so that the scale is standing upright. Keep those in a really warm, kind of dark location, ideally for around 65 to 70 degrees, just to avoid water loss. And over a period of several weeks or around 6 weeks, you'll see those small bulbs will form, and you can discard any scales that have gone soft and get rid of them. Those bulblets can be planted out into pots in order, again, they may send up some vegetative growth, but you're not likely to see them flower. You just want to allow them to build up some strength and some energy over the coming year. Tubers, of course, tubers, the first thing I normally think of when I think of tubers is the Irish potato, because we all do that in school. But other plants that would be tubers include dahlias and caladium. With tubers, what we want to do is just cut it into sections, making sure that we have at least one node on there. You can see the pictures of the dahlia there being separated. You want to make sure that you have those eyes on that tuber, because that is, of course, where you're going to see the growth of that plant coming off that section of tuber. You do want to make sure, when you cut tubers apart, and I do this with seed potato, when you do separate out those eyes for planting, it's a good idea to let them sort of sit for about a day. Sometimes it takes 2 days, just to form a little bit of callus tissue over that wound where they've been cut apart. And that will ideally reduce some of the rot problems that you may have with that tuber in the soil, and give you a little bit better success working with that. Dividing rhizomes is really straightforward. A rhizome is a modified stem that grows just right at or right below the soil surface. You can separate that out into nodes. Really good examples would be canna and irises. Cut away those individual new rhizomes from the mother rhizome, from the oldest part of that root. And you really want to inspect this and make sure that all the sections of root that you've cut away are really firm and really healthy. Throw away any that have any bad marks on them, or if they feel soft, as well as the older part of the plant. It's also a good idea when you do this to trim the foliage back. That is just going to reduce water loss and give you a little bit better chance of that new root system being able to manage the growth of that plant. But with these, they can be planted back immediately, very successfully, and is a very simple way to propagate these plants. Clump divisions usually used in ornamental grasses. To divide these, first of all, we want to go ahead and cut that foliage back to about 4 to 6 inches, and then ideally dig the entire clump up from the ground. And then you want to just use a good sharp tool to separate that clump. And recommendations online are amazing. They suggest using a good sharp knife, and if that doesn't work, to use an axe. And there's some wonderful pictures of people separating the clumps with a standard fireman's axe, or a tree cutting axe. And I've even seen some recommendations that people use chainsaws to separate out these clumps. Hopefully you have an easier time of it. Most of my experience has just been using a really good sharp knife has been effective for that. A lot of times when we have ornamental grasses, sometimes the center will kind of die out. So we want to make sure that we get rid of any of that dead tissue and just discard it. Make sure that we have good root system and good shoot growth on all the sections that we're putting back in. And again, we can put those divisions right back into the ground and really spread that plant throughout our landscape. Last kind of division I'm going to talk about is sucker division. A sucker is a shoot or a branch that's growing from the base of the plant. And this is a really simple form of division because that sucker really has done a lot of the work for you. It's got a good shoot coming up out of the ground. It's developed a root system. So what we want to do is we want to loosen the soil around it, carefully expose those roots so we're not damaging them, and then just lift out the sucker and those roots, doing the best job we can not to do any damage to the parent plant in the process. Cut that away from the plant. Make sure that we're removing any of, we make sure we have all those good healthy roots there. And it's generally a good idea to trim that sucker back. We're both removing the main root or the main stolon and leaving the fibrous roots, as well as reducing the shoot growth by about half. That just reduces the water demand on the roots of that sucker. And we do want to make sure, of course, the best plan is to put these in pots and really monitor them, make sure that we're giving them just the amount of water that they need in order to promote their growth. Moving on to the other kind of propagation that I'm going to discuss for today, that is layering. And layering uses parts of the plant that they're still attached to the parent plant to form roots. And oftentimes we will see some plants that just do this naturally. And this is a very effective way to propagate plants because it really minimizes the amount of stress that the plant is going to be under and has very good success as a form of propagation. Oftentimes we are going to be wounding the area of the plant where where that new root system is going to be formed. And one thing that I always want to make sure that I note when I talk about that is that sanitation is going to be very important. We want to make sure that we use a very clean, very sanitized tool, particularly if we're going to be layering more than one plant in a day. We want to make sure that we wash off our tools between use and apply something like a vinegar solution or a bleach solution in order to prevent any possible contamination. We also want to make sure that whatever rooting medium that we're going to use provides good aeration and really good constant supply of moisture. Again, peat as a material is very useful for this because again we're looking for something that is going to have good water holding capacity but also really be effective to allow air movement into that root system. So the simplest form of layering is simple layering. We often will see this used with plants like climbing roses or boxwoods, azaleas and wax myrtles, and quite a few others. And in this, we're going to bend a low-growing flexible stem down to the ground, cover that with soil, ideally leaving about 6 to 12 inches of that branch beyond that position. And it's often useful to stake that into a vertical position just to control its growth and make sure that it stays really stable there in the ground. Oftentimes, it is useful to wound the lower side of that bent branch. That can promote the formation of roots in that section. And we want to make sure that we check that for moisture, make sure that that area of the soil is staying really good and has plenty of water. And we want to check periodically every couple of weeks to see if we have root formation in order to see when we can separate that plant. Do keep in mind that layering can take time, and it's better to be patient than not, as oftentimes it may take more than one season before that layer is really ready to be separated off from the parent plant. Once it is really well rooted, you can simply cut the section of the branch and then gently take that layer back out of the soil, trying to preserve its root system, and generally move it into a pot. One thing that I have seen done very successfully is to rather than take that branch and push it down into the native soil, you can actually put that down into a potting medium and essentially layer it into a pot, which can be very convenient and a little bit easier to work with. Compound layering is very similar to simple layering. It's essentially the same thing, but it's something you'll see done with vining plants, things like pothos or wisteria. Grapes would be another good example of something where you could use compound layering. I don't know if I can stomach the idea of propagating wisteria simply because I've been trying to get rid of it for quite a while. But another plant where that does work. So very similarly, the only thing different you're going to do here between simple layering and compound layering, sometimes this is also called serpentine layering. So you're bending that stem down into that rooting medium, but then you allow the stem to continue back up above the soil, and then again another section down into the ground, and you can do that over and over again down the length of a vine. Again, wounding that underside can allow for a little bit better root growth on those layers. Mound layer, another form of layering that we would use. This is something that we would tend to see with heavy stemmed or closely branched shrubs. And I tend to think of this as sacrificial layering. Because we do cut the plant back to about an inch above the soil in the dormant season. And then those dormant buds start to produce new shoots. You've probably seen this if you've tried to remove trees or remove plants from your landscape and left the stump there. Often new shoots will grow out of that stump. And that is precisely what's happening. You want to mound soil up over those new shoots as they're growing. And then roots will develop at the bases of these shoots, and then you can pull that soil away as we get back down into the dormant season again. You can separate those rooted layers from the base of that plant, and that provides those the propagated layers that you're looking for. Air layering is the last form of layering I'm going to talk about. And is a really excellent method of propagating a wide range of different plants. Works very well on azaleas. I'm most familiar with it in air layering camellia, but also works in magnolia and oleander and holly. So it's a very effective way of propagating these plants. You want to choose an area just below a node and about a foot from the end of a branch, and remove the leaves and twigs on that stem, 3 to 4 inches on either side of that point on the branch. Ideally, we're going to do this in the spring on shoots that are produced during the previous season, or we can do it in mid to late summer if we have shoots from the current season's growth. So we can really go either way on that. You can also use this as a way to propagate large overgrown houseplants, things like rubber plant or croton or Dieffenbachia can be propagated by air layering. One thing that is important is the method we're going to use for air layering is going to be a little bit different between monocots and dicots. I just want to take a second as a reminder that monocots, when we look at them, their initial growth is going to have one seed leaf. That's where the name monocot comes from. But when we look at the leaves, it's probably the easiest way to differentiate them. Monocots are going to have leaf veins that all run parallel to one another, whereas in dicots, they tend to have, of course, they have two seed leaves, two initial leaves that form out of the seed, and then the leaf veins are going to be more net-like, or kind of look like a miniature plant, miniature tree of veins on the underside of on the leaf. So do need to be conscious that we are going to approach this slightly differently. For air layering monocots, we want to make an upward about 1.5 to 1.5 inch cut, about 1/3 of the way through the stem. So you can see a picture of that in the upper photograph there, and it's we want to hold that cut open. I would recommend using a toothpick or a wooden matchstick just to kind of prop that cut open to expose that area. And then you want to surround that with good moistened sphagnum moss. You don't want it to be too wet. You want to really thoroughly wet that sphagnum moss and then kind of squeeze the water out of it, because you don't want too much water. So you're going to wrap that around that area where you've created that wound, and then wrap plastic around that, and then fasten that securely with twist ties or electrician's tape. Some people use aluminum foil as a just because they don't have to use fasteners in that case. But we want to make sure that we're not either allowing moisture into that area, and we're not allowing that moisture to come out. So we really want to keep it good and constant there for that layer that we're making. There are a number of commercial products that I've seen that are essentially plastic shells that you can place around the area that you're air layering. I haven't had the experience, but they seem like a pretty good idea just as a simple way to approach this. You do want to make sure that if you are putting a layer in an area where you have full sun, that can cause it to heat up or dry out. So it's a good idea to cover it with an opaque plastic if you're in that area, just to prevent any issues with excessive sunlight or excessive heat. You can check back for roots after about 3 weeks. Different, plants in different conditions or different species may take, a shorter or longer time in order to form roots. But ideally, you should be looking at root growth between a range of about 3 to 7 weeks. Once you have really good developed root system, just cut that stem below the medium, and you can just go ahead and pot that layer. And you do want to make sure that you provide really good shade and moisture for that plant until it becomes well established. With dicots, the process is just a little bit different. We want to start off with a stem that's about a pencil diameter as a good rule of thumb. And what we want to do, kind of all of the same things for using monocots, but rather than having that cut upwards, we want to remove about a 1 inch ring of bark from the stem, leaving that inner woody tissue exposed. We do want to make sure that all of the cambium tissue is removed. Occasionally, when we do this, we'll have callus tissue that will actually join across the cut, and we don't want to see that because that's going to prevent the root growth that we want. And oftentimes, we will apply a rooting hormone similar to root tone or one of the auxin-based rooting hormones that is going to help produce roots for this layer. And then you're going to wrap it just the exact same way as you would with monocots. The time it's going to take is roughly the same in that same 3 week to 7 week period. Just come back and check and make sure you have root development. Cut below below where you're creating a layer, and go ahead and put that into a pot, and you should have good success getting that started. So unfortunately, I am running short on time for today. So what I am going to do is I'm going to cut this presentation into 2 sections. And in next week's presentation, I am going to discuss cuttings and grafting